Hey, what's going on? It's January 7th, 2022, and it's a great day to learn more about the immune system, of course, because it snowed outside. And I'd show you a picture, but the sun is starting to melt it all. But yeah, means that there's going to be more colds and more reasons to think about how your body prevents the various diseases that take advantage of the cold weather from affecting your system. So, but not going too much into that because that'll be a whole different video later on. I'm doing a little bit of a deep dive today. Well, not even a deep dive, but kind of a deep dive into the B cell receptors. Okay, so we know about the B cells, right? B cells are one of the primary cells of the so-called humoral immunity system. Okay, humoral immunity is one part of your immune system, and as I learned not too long ago, humors, as they call it, are referred to. They're another, I guess, older timey word to refer to body fluids. And the humoral immunity or the humoral immune system acts based on macromolecules within your humors, your body fluids. So it's molecule mediated as opposed to the cell mediated immune system. The cell mediated immune system is what most people initially think about when they think about white blood cells and macrophages ingesting pathogens and presenting antigens and whatnot. But the B cells, remember, the B cells are responsible for the antibody production via the plasma B cells, right? So antibodies are macromolecules which bind to antigens, and they do. We went over the different things they do, but they will essentially attempt to halt an infection in their own way by either interacting with the complement system, interacting with macrophages through opsonization, interacting with the pathogens themselves and slowing them down and whatnot. But we should know that one of the important aspects of this system is that the T cell and the B cell interact in a dance, right? An intricate dance where, based on whether the B cells and the T cells in this dance have been have been specified through ingestion of the pathogen and presentation of an antigen or a piece of an antigen known as the epitope, the active region on their MHC class 2 receptors, right? Based on that interaction, the B cells can start to proliferate, right? And so as the B cells proliferate, then the T cells also proliferate, and then they start to produce more antibody factories, and the cascade continues. So the thing is, one thing that we should know is that B cells, being immune cells, have MHC class 2 receptors. Remember, that's how immune cells talk to each other: T cells to B cells, B cells to, or one way that they kind of talk to each other and kind of activate each other, and B cells to macrophages, etc. But B cells also have their own receptors called B cell receptors. B cell receptors are actually, the whole system is quite complicated, but the B cell receptor is basically a membrane-bound antibody. So that's what it's called, a membrane-bound immunoglobulin, another name for it. And you can tell by looking at it right here that these B cell receptors look like antibodies stuck in the cell membrane. Okay, and these antibodies, obviously the system is quite complicated, but if you zoom in a little to the actual this image here, you can see that most of the shape of the antibody is the same. They have the variable regions, and then they have the constant regions, and the difference is that they have these cytoplasmic domain, which are in transmembrane domains, which anchor them into the phospholipid bilayer of the B cell, the membrane. And then what happens is, through pretty complex signaling process, these immunoglobulin A and immunoglobulin alpha and immunoglobulin beta, we have to be very specific, which are also bound to the membrane, actually perform the signaling. And what is the signaling? What is the point of the signaling? One, when the antigen binding occurs in the variable region, right, via recognition of the epitope, epitope-paratope interaction, the a signaling molecule, a cascade of events occurs, which involves this immunoglobulin alpha and beta, right? We can look into this more. See IgA and IgB. It looks like these legs, upside down legs. And there's so much going on here, but just to note that these two areas are called ITAMs, right? These ITAM regions, right, interact with something called a Lin kinase, and this Lin kinase phosphorylates these regions, and then a whole cascade of events occurs, which is more of intermediate immunology to understand how that works. But essentially what happens is that goes all the way down to signals go all the way down, this antigen signaling molecule goes all the way down into the nucleus, and these B cells get activated that way. And B cell activation means there's things called cyclins which promote, and these are general molecules, they promote the cloning of the B cells, right? Because remember, the cloning of the B cells is what is important, right, to the B cell. And remember, that's how the talking occurs, right? The T cell will talk through to the B cell, and B cell will talk to the T cell, right? And once that occurs, then all this chemically happens within the B cell. But I mean, most people just know the systems level version of how this works, but that's fine. So yeah, and then antibodies are produced through the plasma cells when the B cells are matured in the lymph nodes. Remember, all of this is happening really mostly in the lymph nodes, in specific regions of the lymph nodes. If you think about the lymph nodes, as I originally did, and just think that they're these places where a hodgepodge of events happened, it's actually quite complicated. And I've heard lymph nodes being described as the army base of the immune system, because that's essentially where plasma cells are created. That's where the B cells talk to the T cells. That's where T cells have their own region in the lymph nodes. So it's subdivided. It's not just a hodgepodge, and it chemically can, as usual, get quite complicated. So again, there's communication between several ways between T cells and B cells. There's binding through MHC2 and the T cell receptor, and the antigen locked into the MHC type 2. Then there's cytokinetic communication. And then one last thing I want to talk about is that essentially there is, and we'll get into this later, but there's a term that I learned recently which is quite interesting to me, and it makes sense, is that these cells, the antibodies, the variable regions of the antibodies undergo something called somatic hypermutation. Okay, a fancy sounding word, but somatic means only works or only applies to somatic cells, non-germ cells, and hypermutation refers to changes, incremental changes and quick changes to the DNA which occur to allow the antibody to keep up with a viral sequences of viral changes, right? Like, so viruses mutate quickly. Even if they don't become a whole new strain, they still mutate quickly as they're progressing through an infection, right? And then their spike proteins change it and whatnot. So essentially, these antibodies have to keep up with those changes, and antibodies are antigen specific. Remember, so the antibodies refer to the macromolecules, the Y-shaped macromolecules that are free floating, and then also the B cell receptors as well, right? Because remember, the B cell receptors also have to have this antibody work that they're doing, right? Like of recognition and whatnot. So the DNA has to change, and the variable regions have to change, and they change quite quickly, and then a whole entire cascade of events, usually that's what it's called, through several different enzymes and kinases occur such that these regions change to adapt to the new antigen. That's the very high level, but then you have to start going into things like antibody affinity and whatnot, and it becomes quite complicated. So I'm gonna stop there because this video is already 10 minutes about. So yeah, I'll leave you to the rest of your day to think about B cells and T cells, because that's all you're gonna be doing today, I'm sure. Right? All right. Well, see you around.